In this slide, I am sharing some extra information about gram-positive bacteria and gram-negative bacteria. That are the two types of bacteria. And the name gram-positive and negative comes from Hans Christian Gram, who developed the staining technique to distinguish between the two types of bacteria. The staining technique is a technique in which cells, here bacterial cells, that are normally transparent are immersed in one or more colored dyes that are called stains to make them more clearly visible through a microscope. So we are going to study about the staining technique in brief. And apart from this technique to distinguish between the two types of bacteria, even a peptidoglycan layer. Now peptidoglycan is a polymer of sugar and amino acid forming layer outside plasma membrane of bacteria with cell wall. So this peptidoglycan layer is present in gram-positive bacteria, whereas it is also present in gram-negative bacteria, but a very small layer of peptidoglycan is present. So this is one difference between the two types of bacteria, that is presence of thick peptidoglycan layer in gram-positive and thin layer in gram-negative. Now the next one is the staining technique. A counter stain is a stain that makes cells or structures more visible when not completely visible with a principal stain. So there are two types of stains, that is principal stain and a counter stain. Crystal violet stains both gram-positive and gram-negative organisms. So crystal violet is a principal stain, and when the bacterial cells are stained using this stain, this crystal violet, both gram-positive and gram-negative will take up the color of the stain, that is violet color. So you cannot distinguish between the two types because both have taken up the violet color. Treatment with alcohol removes the crystal violet color from gram-negative organisms only. So what is done? Next, the bacterial cells will be treated with alcohol, and what happens is that the crystal violet color from gram-negative organisms is lost, whereas gram-positive are violet, whereas gram-negative lose their violet color. Now we make use of safranin as a counter stain. So safranin is a counter stain and it is used to color the gram-negative organisms that decolorized by alcohol. So the color of safranin is somewhere from pink to red, and by using safranin as a counter stain, we can see the gram-negative organisms clearly under a microscope, and as they had become decolorized by alcohol. And another interesting thing, that is gram-positive bacteria do not take up the safranin stain. So this is what are the two differences between gram-positive and gram-negative bacteria. Now in this diagram, it can be clearly seen that how the peptidoglycan layer, which is a polymer of sugar and amino acids, is a very thick one in case of gram-positive, whereas in case of gram-negative, you can see it is only a very thin layer. So peptidoglycan layer is thick in gram-positive and thin in case of gram-negative.